So, as we've seen, genetic analysis of experimental organisms like Mendel's peas allows for controlled crosses to be made. A controlled cross is simply where an experimenter is choosing the phenotype and/or genotype of the individuals used in a cross. Now, this type of analysis is not always possible. Genetic analysis of humans or other non-experimental organisms requires a different type of approach. One where essentially you're looking at crosses that have happened and interpreting them. And one approach to use with this type of situation is something called pedigree analysis. Pedigree analysis is simply an analysis of family trees, essentially, and using these to deduce inheritance patterns. So pedigree analysis makes use of crosses that are already available, and it uses them to deduce a number of different things. The first thing that one needs to deduce from a pedigree is the mode of inheritance for a condition. And we'll talk about this in a moment, but there are 4 different possible modes. A condition might be recessive to its corresponding wild type, dominant to its corresponding wild type, and then the location of the gene that's involved in a particular condition might be on the X chromosome, on a sex chromosome, and be sex-linked, or it might be on an autosomal chromosome, a non-sex chromosome. So that gives us 4 different possible modes of inheritance: autosomal dominant, autosomal recessive, sex-linked dominant, sex-linked recessive. Once you've determined the mode of inheritance within a pedigree, the next step is to assign genotypes to as many individuals as possible. Now, it may not be possible to fully assign genotypes to everyone in the pedigree, simply because some genotypes might remain somewhat ambiguous, and we'll see examples of that in a moment. Once you have the mode of inheritance and genotypes assigned, then you are able to use these lines of information to assess probabilities. So probabilities that certain individuals will be affected with a condition, probabilities that certain individuals might be carriers, i.e. heterozygous for a condition, or perhaps the probability that an individual or a couple might have an affected child, for example. So the 4 inheritance modes that we just mentioned: autosomal recessive, sex-linked recessive, autosomal dominant, and sex-linked dominant. And it's only ever one of these 4 possibilities. Okay. The first decision, as we mentioned, that you need to make is recessive or dominant to the corresponding wild-type allele. So maybe one thing to say about recessive or dominant: again, recessiveness and dominance is not an intrinsic property of an allele. So in this case, we're talking about the allele that's responsible for the condition, and we're talking about that allele's relationship to the corresponding wild-type allele. And when we say a wild-type allele, what we mean is the allele that provides the normal function, the non-disease state for that particular locus. Okay, once you've decided if a condition is recessive or dominant to its corresponding wild-type allele, the second decision is to determine whether or not the locus where this allele or these alleles reside is autosomal or sex-linked. Okay, as we've just been mentioning, wild type is a normal allele for a locus, and for a genetic condition this means the non-disease allele. Wild type is actually one of those concepts that can be a little fuzzy at times. It is possible, and we'll see this in later lectures, that there can be a number of different alleles present in a population, and none of these alleles are necessarily what we would consider a mutant allele. For example, the different hair colors in humans. The alleles at that particular locus responsible for the different hair colors, they're different alleles, and whether one chooses to call one of them wild type and the others mutant alleles is really somewhat of a matter of taste. In the cases of genetic diseases, it's straightforward. The disease-causing allele is the mutant allele, and then the non-disease allele is the wild type allele. A good point to make again is that any mutant allele that you care to define will always have a corresponding wild type allele. So as soon as you define something, just call it little a, for example, for a recessive condition, as soon as you define that little a, you are also instantly defining a wild type allele of the A locus, say that we would call uppercase A. So if you can define a mutant, then there is a corresponding wild-type allele. Pedigree analysis has a specific symbolism that you need to become familiar with. Circles represent females, and squares represent males. A bar connecting a male and a female indicates a mating, or a marriage if we're talking about humans, and the vertical bar descending from that horizontal bar indicates offspring of that particular mating. Individuals that have a filled-in symbol indicate that they are affected with a condition. And please, it's affected with an A and not infected. Medical microbiology is going to be all about infection. In Biology 371, we're specifically talking about affected individuals, people who are affected with a genetic condition. A diamond-shaped symbol represents an individual for whom the gender is not specified. This doesn't mean that the individual has ambiguous gender, but it means that for the purposes of this pedigree that their gender is not specified. There are some other symbols that you will see in other pedigrees as well. If there happen to be two lines connecting a male and a female, that's an indication that it is a mating, but a mating among people who are related to each other. So that could be more distant relatives, human relatives that are married to each other, or in the case of using pedigrees for animal breeding, say pedigree dogs or show horses or something along those lines, oftentimes there will be relatives that are mating with one another, and that will be indicated with a double bar. The other thing to notice is that pedigree generations are denoted with Roman numerals, so Roman numeral 1, 2, 3, and so on. The other thing to note is that individuals within a given generation also have numbers, but they're not typically represented on a pedigree. So this would be individual Roman numeral 1 - Arabic numeral 1, so 1-1, 1-2, 3, and so on. This provides a very convenient way to talk about individuals within a pedigree. For example, we would say that individual 2-1 here and 3-2 here are affected individuals. It's a very convenient way to call out a specific person in a pedigree, and you can use that in your answers as you're writing out your answers for pedigree questions. It's easy. It's a good way to refer to individuals specifically by their number.